Hello again. We're going to talk about DNA and a little bit more about DNA technology. So recall our situation here with Jack the Ripper. And back in when these murders were committed there, we didn't have all of this DNA technology. But now we have the ability, right, to go in at a crime scene and gather blood and skin cells, and if there was any biting saliva or tissue under the nails. It's really common that victims will scratch at the perpetrators, or hair or semen if it were a rape victim, or whatever. There's lots of different biological material that's left behind, and we now have the ability to go into those cells and then compare those biological samples to samples from both the victim and the potential suspects. So I want to remind you that if we go into DNA and we want to make multiple copies of DNA, remember that the body does this naturally with a few main ingredients, right? You need to have a helicase that unwinds the DNA. You need to have all of these free nucleotides that are then going to be added to the new strand that's being built. You need to have a polymerase that knows how to match up the different nucleotides and build the strand that's being built. Right? There was also the primase that was laid down. That's when you first start the copying process. So there's lots of different types of ingredients that are part of replication. And when we do this in the body, we also have different. When we do this in the lab, right, inside of a test tube, we can mimic what's happening in the body. So look at the main ingredients that go inside these test tubes. You need to have a little bit of water and buffer, and so that gives you some volume to work with. You need to have DNA, the minimum amount is you need at least one single strand of DNA. And this is why you could technically get enough DNA from just one single cell. But usually even in a small sample of blood, you can get hundreds of cells of cells, and therefore hundreds of copies of DNA. You need to have primer, and I'll show you what this is in just a moment, but this helps initiate the process of copying. Nucleotides, so you need to have all these free A, G, C's, and T's that are floating around in the liquid so that they can then be added to. And then you also need, of course, the enzyme polymerase, which is going to be the enzyme that goes through and runs along the template strand and adds the complementary nucleotide, right, to build the new strand the new complementary strand of DNA. So this happens in a machine called a thermal cycler that looks something like this. You lift the lid up, you put this test tube inside, and then you have this environment heat up and cool down with these different ingredients that we've added. And so what happens is, notice we didn't add a helicase. Instead, we heat DNA up, and we know that when DNA is heated, it denatures, or in other words, it separates. So because we don't have a helicase, we can just heat the DNA up and it will separate. So we go from then we'll bring the DNA back down to bring the temperature down a little bit, and we'll allow the primer sequences, which are just short strands of DNA, will bind to the complementary, right, base pairing of the nucleotides on the template strands. And so they bind to that, and then you do a little bit more of a temperature change, and this brings the polymerase into action, and the polymerase starts at the primer and then continues to build forward, bringing in the DNA, and having all of these free nucleotides are then being added based on whatever the complementary nucleotide is on the template strand. And so this way you build, right, copies of this DNA. So essentially in the first cycle you've got 4 copies. By the second cycle, you've now got 8 copies. By the third cycle, you've got 16 copies, and by the fourth cycle, you got 32 copies. So this is an exponential process, and you can think of it this way: if you do 30 cycles, then that's 2 to the first. So that's 2 billion copies of DNA. So this is, you can quickly, from a very small amount of DNA, get lots and lots and lots of copies. And then you can do different analyses with these copies once you have them, which is what's done in DNA technology and forensic science. When they only have a little bit of DNA to work with, they can copy the DNA using polymerase chain reaction, or PCR. Now, we want to talk about the ways in which you can visualize the DNA. And one way is to just sequence it, and you can actually just read the letters. And we talked a little bit about that with the STRs. That's one way that you can do this, is just actually read, the number of copies. But you can also do it this way, where you do a visual read, and so you use a gel. So this is an agarose gel, so think of it like gelatin. You've got like this cube of gelatin, but you have put in a mold that has these little wells. So there's like these little wells that are empty, these little holes. And you can take the DNA, and you can put the DNA down inside of these holes, and then you can turn on a charge across the gel, so that one end becomes more negatively charged and one end becomes more positively charged. Okay? So you can load DNA, and then when you turn on that charge, DNA will migrate through the gel, and DNA migrates from the negative end towards the positive end because DNA is overall negatively charged. So as we look at this, again, here we've loaded DNA in, and then we can run the DNA out and it's going to migrate from the negative to the positive, and it migrates though at different speeds. The shorter fragments, the shorter length DNA fragments, migrate faster through the gel, and the longer DNA fragments migrate slower through the gel. So you get this nice spread out pattern. And in fact, you can then start to compare, one person's DNA spread relative to another person's DNA spread, and we call that the DNA fingerprint, because you have these very specific bands that spread out across the gel based on the charge as they're moving through the gel, based on the size of the DNA fragments. Now, if we don't cut the DNA up, though, into different sized fragments, then of course it all just moves, in one big chunk together. So when we're going to make a DNA fingerprint, we need to cut DNA up into fragments. And we actually have these molecular scissors that help us to do this. And it turns out that these molecular scissors find these DNA palindromes that are written into the code, where, for example, here you see it's CCGG in one direction, and in the other direction it's CCGG. So this is like race car, right? It's like a palindrome. It's the same forward and backwards. And so these molecular scissors come in, and they can cut DNA along these. And so if I add to my sample of DNA that I have done polymerase chain reaction on, I've done PCR, so there's millions and perhaps billions of copies of these strands of DNA. If I then add these molecular scissors that are going to go in and cut the DNA everywhere they see, CCGG, right, this pattern, it'll cut it, then you'll end up with one strand from here, a second strand that's this middle strand, because it also is going to get cut near the end. And notice that these sizes are slightly different. X is the largest length DNA strand, Y is the next largest, and then, W is the next largest, and then Y is the smallest strand. But it's also possible that, remember that everyone's DNA is slightly different. Some people have slightly different variations across their DNA. And so it's completely possible, it's completely possible that someone maybe doesn't have the CCGG pattern, but rather they have the ACGG pattern. So the molecular scissors will not be able to cut the DNA right there. So instead of cutting the DNA into 3 pieces, for this person, the DNA would be cut only into 2 pieces, into a fragment of size Z, but then you would still get the cut, because at this particular part of the DNA, they share the exact same pattern as the other person, and so you would still get fragment Y. So again, now we've cut our pieces. Now we can take those, load them into the gel, and then run them out on the gel, and this is what we would see. We would see that the crime scene DNA, again, was cut into 3 different pieces of length X, W, and Y. Recall that Y was the smallest, and so it traveled the most distance, right, across the gel. W was the next smallest, and then X was the largest for the crime scene DNA. But recall that the suspect DNA was only cut into 2 pieces, and in fact, essentially, X plus W equaled the length of Z, and so Z didn't travel as far as X. It's even further up in the gel, and then Y traveled the exact same distance as the Y band that traveled in the crime scene DNA. And this is how you can compare biological samples from crime scenes to biological samples from suspects. If they match, then that's evidence supporting that the suspect left a biological sample at the crime scene. If they don't match, then it's evidence that that suspect was not there, or at least that biological sample is not of the suspect. So these molecular scissors then are called restriction enzymes, right here, restriction enzymes. And the analysis of cutting DNA up into different lengths is called restriction fragment length polymorphism, okay, or RFLP. And so here is another example of a restriction enzyme called EcoRI, okay, EcoRI. And if we look across this stretch of DNA, so notice we're looking at both complementary strands here, you need to look then for a palindrome of the pattern GAATTC. So if you quickly look across this, I'll give you just a moment. You look across all of these letters. Can you see anywhere that this would be cut up? And if so, how many are there, and therefore how many resulting strands would there be? So we look across this, and this right here is the only place where you have GAATTC in one direction and GAATTC in the reverse direction. And so the correct answer to this question would have been 2, because this piece of DNA would be cut into 2 different fragments. So taking all of this into account, we can then start to solve not only forensic science, like a crime scene, but we can also do like paternity testing. So for example, here I've got a picture of a gel that we've ran out. Of course, this is just a cartoon here, but of a child and a mother, and then 2 potential fathers. So the question is, who is the father of the child? So as you look at that, at the DNA fingerprints of each of these 4 people, you should be able to figure this out. Well, if we look at this, it looks like the father must be Larry, because the child has this DNA fingerprint where they have a band here, a band here, a band here, and a band here. Well, it looks like the mom for this particular, for this DNA, perhaps her DNA never gets chopped up with this particular enzyme, and so she only has one band, and the child also has that one band. However, the DNA that came from the father happened to get cut up into 3 different bands. 1, 2, 3, 1, 2, 3. Notice that Bob has a band that does not appear in the child, and Bob has a band over here that does appear in the child. But the best match for this particular test would be Larry would be the best potential match for the father of the child. So do you remember Jack and Jill? Well, we finally got the paternity test back from Jack and Jill and Donald and Daisy, and here are the results. So what do the results tell us? Look at that real quick. This is Daisy's, this is Jill's, and this is Donald's. So what do we see? Well, Jill is definitely the daughter of Daisy because she has both of the bands of Daisy. Does Jill have the bands of Donald? That's really the question that we're asking. Well, that one doesn't match up. They're different ones. Those don't match up. Jill's got 2 here, and they're not present in Donald, and then Donald's got 1 way down here. So do they match up? No. So the answer would be they are unrelated. So it's all a happy ending for Jack and Jill. They can get married. Donald is not the father of Jill. So I hope that puts you all at ease.